I would now like to welcome Naveen Vankadari, who is joining us from Australia, and they will be giving a talk titled Structural Mechanism of SARS-CoV-2, Entry and Activation of Spike Glycoprotein by Engaging Unique Host Factors and Potential Antiviral Interventions. So if you can get your presenter. It looks perfect. You can proceed when you're ready.
Can you hear me?
Yes, we can hear you. Please proceed.
Great. Foremost, thank you very much for giving an opportunity to present in the a great conference, a symposium where to interact with renowned people and sharing the knowledge. And this looks like a third talk on SARS-CoV-2 virus, but I'm try to ensure that I'm going to talk a little bit different at molecular level and atomic level, what's going to happen for the viral entry in the human cell. We all know that presumably bat-originated coronavirus has infected more than 430 million people worldwide and causing a range of illness and ultimately the COVID-19, the disease. And despite we have several number of different types of vaccines available in the market, and as well as most of us are being vaccinated, we're still looking for a potential therapeutics to fulfill the therapeutic gap and also to treat the COVID-19 or SARS-CoV-2 infected patients. And also it is an analogy to develop new drugs to combat any other variants as well. And so today we saw that the number of mutations, including the Omicron, which has been derived. If you look at the 2019 December in the start in the left-hand panel, so the right-hand panel of the genetic variations or the mutations are happening, it's tremendously increasing in every bit and region of the chromosome or the DNA region or RNA region of the SARS-CoV-2 virus. So that has also resulted in the changes in reversal and changes in the genome of the virus has also resulted in the cause of the Omicron. And I will come to that, how the Omicron is also working on and whatsoever. But ultimate path of a viral entry is mediated mostly by the receptor-mediated entry or the direct endocytosis. But whatsoever, it uses the host machinery to make the new particle. And like what we see in the textbook diagrams, like a 1 or 2 viral particles are attached on the host cell surface, but it doesn't look like that. But each cell is attacked with hundreds and thousands of viral particles. It's more complex and more challenging on the surface of the real human cell or in a lung virus. So when we look at the TEM or an electron microscopy images, when viral particles are entered in the human cells, they're not just one, probably 10 or 20 has also been entered into in a one shot. So then it is it's difficult to understand how the junction of the virus or what is happening at the junction of the virus and as a human cell. So what is the mode and the mechanism of viral entry is happening so that we can target that entry or the junction and we can possibly cure the disease as well. So if there's a case and we have to understand what is happening in this junction of the virus in the human cell, we need to look into more deeper sense using an atomic resolution or at the atomic level or protein and protein interaction level. And that's the way we have to target any drugs or any vaccines or antibodies or anything to do. So as in a simple terms for a non-structural biologist to tell, an amino acids are aligned in a chain and fold that causes a peptide and folds into a different conformations, and that makes a polypeptides. And a tertiary conformation of the polypeptides makes a molecules are functionally active enzyme. So which we use, the technique is in a crystallography or electron microscopy, where we crystallize this kind of proteins and diffract with an X-rays and then get the electron density which is present on the surface of the protein, and then solve the structure of a protein or a human or a virus, whatever it could be. So in the early 2020, we were the first in Australia, at least to say, in the competitive race, we identified the key differences between the SARS-CoV-1 and the 2, where the N-terminus region of the SARS-CoV-1 has diversity. and as well as the presence of a furin cleavage site in the SARS-CoV-2 virus, and solve the structure of a closed and open conformation of the SARS-CoV-2 spike protein that is important for engaging the host receptors. In the meantime, in Nature paper, the other groups has been published that the SARS-CoV-2 engages the human ACE2 receptor for entry. So, but if that's the case, then we have to understand what is the sugar or a camouflaging pattern across the SARS-CoV-2 spike protein, because most of the virus particles, like in a spike proteins, are shielded within a sugar called as a glycoprotein. That's why they are called as a name as a spike glycoprotein. This glycone or a sugar acts as a camouflage where it restricts most of the antibodies to target. So understand the camouflaging pattern is also important to design the specific antibodies which is not glycosylated. And that's what we found that SARS-CoV-1 and 2 were the differences in the glycosylation patterns, and also proposed a new receptor for SARS-CoV-2 entry in the humans, that is a human CD26, or also called as dipeptidyl peptidase, which received more than 500 citations and being considered as the top 10 article of the year in 2020 as well. So, which means like the SARS-CoV-2 does engages the other receptors like a CD26 and others along with the human ACE2 receptors. It could be quite possible, because if it's ACE2 is the only receptor for SARS-CoV-2 entry, if we could have effectively blocked the ACE2 receptor for the SARS-CoV-2 infection, then we could have completely eradicated the infection, or we can cure it. We don't need to struggle till now. So the virus does evolve and engages different kind of host receptors for subsequent entry as well. And to understand the diversity of the spike protein, we need to understand how it functions. So towards that, we perform the electron tomography to understand how the real SARS-CoV-2 virus looks like. So when we image the SARS-CoV-2 virus in the tomography, where the Z section is taken from all angles, so we does notice the SARS-CoV-2 in different conformations, like a closed, open, and a fusion related conformation. So which seems like the region in the adhere to the virion is more firm compared to the region which is involved in interacting with the receptors. So targeting this region of involved in the human receptors is most likely not that successful. Then something we have to target in this region where it looks like a little bit of stable in the trimer region. In that regard, we surprisingly identified human influenza virus has a conserved region with the COVID-19, and a drug called Orbidol can effectively block the human influenza virus. Then we have a hypothesis that this drug can also cure COVID-19 or SARS-CoV-2 infection. And we structurally see that, yes, it does bind. And in cellular experiments, we notice that the Orbidol can inhibit the viral entry into the in the cell lines of, but then the question is, how does it binds? There's a mechanism of interaction. So towards it, we solve the structure of an SARS-CoV-2 spike protein in complex with the Orbidol, where orange color Orbidol is sitting in the junction of the spike protein. So let me show you in a movie, so it will be very easy to understand. The spike protein is a trimer, as we all know. So monomers are combined with individual monomers joins together become a trimer. But the Orbidol sits into the dimer interface or a trimer interface of the protein and disrupts the interaction interface. When the two proteins of the primer is not stable, then dissociates. That causes a weak interaction or stability of the spike protein can reduce the infection. This made us to, it's been highlighted in different media saying that we identified the mechanism of Orbidol in curing the disease, but this is all good, but how true in real patients? So in that regard, we first collaborated with the Wuhan Pulmonary Hospital in China to understand in patients how this drug effectively work on, because that's in a most designated hospital in 2020. So then we just directly collaborated with them. It's sort of a 500 RT-PCR confirmed COVID-19 patients from 3 different hospitals in China, along with the Wuhan Pulmonary Hospital, were taken into the cohort studies and tested for the Arbidol treatment. We surprisingly see that the patients who received the Arbidol in 14 days has shown clearance of the viral infection. So number of the lesion size, you can see a greatly reduced. Along with that, we does compare with another drug combinations like Arbidol with ritonavir or Stalavir, and as well as in combination of both. But a combination of with an Ostalavir has shown much more significant effect compared to the Arbidol alone, but Arbidol was much more impactful. So we also tested the individual drugs as well, Ostalavir, Flavivir, ritonavir, but in combination, or at least the Arbidol was playing an important role. And also we noticed a great decline in the mortality in the cohort of all 3 hospitals as well, thus proving the Arbidol not only functionally blocked the spike protein, but also reduced the COVID-19 infection in humans as well. So as I mentioned, the junction of the spike protein is mediated by a number of factors, which I mentioned is ACE2 and CE26. Another 2 host factors called as furin and TMPRSS. Furin is one of the most dangerous stuff causing for the SARS-CoV-2 infection, which I'm going to come now. So, which is never seen, this kind of an insertion site was never seen in another coronaviruses like a bat, MERS, Pangolin, and SARS-1. But this has been introduced into the SARS-CoV-2 by whatever the mechanisms, we don't want to argue that. But what happens when the SARS-CoV-2 spike protein has an furin? So when the furin is in a human enzyme, is a protease which cleave the spike protein into S1 and S2 domains. So it, instead of making in a single spike protein, it makes in the S1 and S2. But in general sense, if a human protein is cutting the spike protein into 2 pieces, it looks good, but it's not. It's good for virus. So first, to understand how it is going to happen and work, so we first saw the structure of the spike protein in complex with the furin proteases. So, and understanding the mechanism of furin attacking the spike protein region and the mechanism it cleaves. So what does happens is when the spike protein binds the furin of the human, and then it cleaves into the colored region of the S2 region and the gray region of the S1 region. So suppose you think like a thief coming in a Santa Claus dress. As soon as it enters and sell things like a Santa Claus is a thief, but it is want to trigger and cut. But as soon as it enters the cell, it strips the dress and becomes someone new. So it completely camouflages and escape the immune evasion and by stripping the whole S1 domain. So if you look at the top portion of the spike protein, and this one is very different. So that's how it is escaping the immune evasion also, and that's how most of the ACE2 inhibitors and drugs are going to fail. But it is definitely helping in neutralizing, but they are not going to help in blocking the inhibitors if you target the S1 region and the spike protein region, because it's something like ACE2 or other receptors is like a main doors for a viral entry, but thief can enter in several other ways, back door, side, or break, and so many other ways virus can enter as well. And we also see the thermodynamics and as well as a computational molecular dynamics. If the wild type spike protein is more stable, but when it is cleaves, becomes unstable. So that's how it's one of the escaping by shedding some portion of this virus spike protein as well. We also identified a couple of drugs, mostly in amber compounds, which specifically block the furin protease and inhibit the infection as well. And the another key receptor which I want to stress about is the TMPRSS2, which is gaining more attention recently. And the story started with understanding in India, the most of the people who are affected, they're less symptomatic compared to the Caucasian population. Then we were very curious, like why the different ethnicity, there is something is going on for getting an immunity in us. So once we look at, we first took around 1,000 patients, some of them healthy and individual with the COVID-19, and performed the whole exome sequencing of those patients, and found that the patients, some of the patients who are carrying a genetic mutation called valine 162 methionine. The patients who had a mutation in the host genome of the TMPRSS2 valine to cis methionine are less symptomatic or asymptomatic. So we also see in the lung CT scans of those patients, like patients who has a carriers, very asymptomatic and mild, but the patients who do not have are more symptomatic. So which means like mutation in TMPRSS is good for us. So how does it work? So to understand that, we also perform the immunohistochemistry. That mutation has a more expression is there, but it is giving a more stability in the less stability for the protein and reducing the infection. So that for that to understand, we saw the structure of the TMPRSS2 protein in the wild type and the mutant. In simple terms to say, as we see in the top panel is a wild type, where you can see the structure of the TMPRSS2 very stable and rigid, but the mutation causes disruption of the TMPRSS2. The disruption or instability of the receptor of the TMPRSS2 makes a weak interaction with the spike protein and weak receiving of the spike protein into the human cells. That's helping protection against the COVID-19 infection as well. So we also found some of the drugs which can block the effectively TMPRSS2, and some of the clinical trials are still going on. And in early stage, we are seeing that before and after these drug treatment, there is a decline in the COVID-19 or SARS-CoV-2 infection in the patients as well. So that showing some promising effect in the in patients. So with this, we summarize and remodel the mechanism of the SARS-CoV-2 entry and the spike protein processing, where individual monomers of the spike protein assembles to the trimer, which can be blocked with the arbidol, or because arbidol disrupt the spike protein, that can also disrupt its interaction with the human CD26 or S2 as well. So if this step has been escaped, we also can be used as an furin inhibitors like an AMBA compounds, which is involved in the cleaving the spike protein into S1 and S2 domains. And if that step is also blocked, we can use another round of an antibiotics, another round of drugs which are blocked on TMPRSS. So basically, combination of the drugs in a one shot could be much more effective to completely cure the COVID-19 infection in the patients, but may not be in single drug, but it may take a longer time. With this, I acknowledge our university, Monash University, and we come in the Department of Biomedical Discovery Institute, and two hospitals in China, Wuhan Pulmonary Hospital and Tongji Hospital, and the Graduate School of Health Sciences in Japan, and AIG Hospital in India for all of our collaborations and clinical trials, and also the Australian synchrotron for X-ray diffraction, and our own EM facility for other studies. Thank you, and happy to take questions.
Thank you very much, Naveen. That was an amazing presentation. For the questions, if people could please pose them in the Q&A section, and Naveen, if you can just type your responses. We do want to thank you for presenting today, especially all the way from Australia. We know that it's very late there, so we really appreciate your involvement.
Thank you.